Good day, this is Professor Will, CBMD PhD, and today's Thursday, March 17th, 2022. It's 6:53 and 3 seconds, a.m. Eastern Standard Time. Today's research will be on the fetus. So let's hop to it here. The placenta plays active part in transferring vitamin D to fetus during pregnancy. Date March 8, 2022. Source, eLife. The scientists have shed new light on the role of the placenta in managing the relationships between maternal vitamin D and fetal development, according to a new study. The findings demonstrate a complex interplay between vitamin D and the placenta and can help inform future interventions using vitamin D to support fetal development and maternal adaptations to pregnancy. As vitamin D is unable to be produced by a fetus, it must be transferred across the placenta, and this is important for both fetal and lifelong health. Maternal vitamin D concentrations are positively associated with fetal bone growth and birth weight, and these associations continue into postnatal life. The previous work has suggested that maternal vitamin D transfers passively across the placenta, but the current study challenges this idea. Research in kidneys has questioned the role of passive diffusion in the uptake of vitamin D. It has instead shown that this uptake is driven primarily by endocytosis of vitamin D, where the vitamin is bound to the binding protein, albumin, and introduced into the organ tissue cells. Researchers proposed that a similar endocytic mechanism exists in the placenta, suggesting that this organ plays an active role in the delivery of vitamin D to the fetus. And to explore this idea further, the team designed a study to find out how maternal vitamin D is taken up, metabolized, and mediates gene expression within the human placenta. They used a perfusion model involving the use of human placental samples collected from term pregnancies immediately after delivery and placental fragment cultures to study the behavior of the organ tissue. And these methods contrast with cell model approaches of previous research into how vitamin D transfers across the placenta, and to determine the mechanisms of placental vitamin D uptake. The team incubated fresh-term human placental fragments with vitamin D alongside albumin for 8 hours. They then analyzed the gene expression of the fragments using a technique called quantitative RT-PCR. The researchers' analysis revealed a significant increase in the expression of the CYP24A1 gene, which is involved in controlling the amount of vitamin D in the body. In the fragments following incubation, compared to fragments that were incubated with vitamin D only, this suggests that albumin might enable vitamin D uptake. And these findings show that endocytosis may play an important role in the uptake of vitamin D into the human placenta, as previously seen in the kidneys. Additionally, the team demonstrated that vitamin D exposure leads to rapid effects on the full set of messenger RNA molecules, the transcriptome, and the proteins, the proteome, expressed by the placenta. And their results reveal that the underlying epigenetic landscape of the placenta, the interaction between the genes and the environment, helps to dictate this transcriptional response to vitamin D treatment. And this is the first quantitative study demonstrating the active transfer and metabolism of vitamin D by the human placenta, with widespread effects on the placenta itself. The research data are generated from the term placenta only. Additional studies are needed to determine how the findings relate to earlier stages of gestation. And together, the insights from the work and future research will be helpful for identifying potential new options for targeted interventions to improve pregnancy outcomes. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.